So it's my pleasure to welcome Dr. Coleman Lindsley, our invited speaker for today's Blanche Tolman Lecture Series and Yale Cancer Center Grand Rounds. So the Blanche Tolman Lecture Series was established in 2012 by Dr. Marvin Sears. Dr. Sears was a longtime chair and founder of ophthalmology and visual science at Yale, and the lecture was established in honor of his mother, Blanche Tolman, who passed away from AML. This was the first lecture series at Yale dedicated solely to hematologic malignancies, and it is intended to bring to Yale pioneers like Dr. Lindsley that have made major contributions to our understanding of the current trends in hematologic oncology. So a little bit more on Dr. Lindsley. So Dr. Lindsley is an associate professor of medicine at Harvard Medical School and at the Dana-Farber Cancer Institute, where he is also the director of the Edward P. Evans Center for MDS. He received his MD PhD in immunology from Washington University School of Medicine. He did his residency at the Brigham and Women's Hospital. He did his fellowship at the Dana-Farber. And amongst his wonderful activities and leadership in the field of MDS, he is a member of the MDS Genetics Subcommittee for the NIH National MDS Study. He is on the steering committee for the NHLBI Transomics for Precision Medicine group, and he is on the molecular committee for the International Working Group for Prognosis in MDS. His laboratory at the Dana-Farber focuses on the biology and the treatment of myeloid malignancies. His genetic studies have changed and created new paradigms in our field, specifically new genomic models of leukemia classification and of how to understand MDS outcomes after allogeneic stem cell transplant. His laboratory uses a variety of approaches, including mouse and cell line models, to dissect the mechanisms behind genetic cooperation during the progression of myeloid diseases. And he has a very interesting and specific focus on leukemia initiation within the context of predisposition syndromes to myeloid malignancies, as well as studying mutations in the field of epigenetics. So before starting, I think there is a token that we would like to give so that you can remember your time with us.
Thank you.
Welcome.
Thanks very much for the introduction. It's too kind. And for the invitation. It's been a pleasure to see some old friends and meet some new ones, including some of the rising stars in our field just before lunch. And so I'll dive in to talk about myeloid genetics. Here's some disclosures. None of them are relevant for what I'm going to talk about today. I think if I were to simplify down what I'm interested in, it's essentially the origin story of myeloid leukemias, or you can call it the ontogeny of disease. And this is essentially how we get from a normal stem cell that functions properly to a florid myeloid malignancy with failure of the bone marrow organ. And the origin commonly is just aging-related degeneration of the stem cell pool. But we can also have acquired predispositions as well as exposures to leukemogenic agents like chemotherapy and radiation, as well as an increasingly recognized number of inherited predispositions that modify the probability of developing clonal myeloid disease in life. And so it's this origin that I seek to understand, and more importantly, try to understand how that origin influences the outcome of patients, the progression of patients, the response to therapies, and offers us opportunities for development of novel therapeutic approaches. And so to bring us back to the basics, the cell of origin here is the hematopoietic stem cell, which functions normally with its dual roles of uh, I wouldn't say perpetual self-renewal, but ongoing self-renewal throughout life, and its capacity for multilineage differentiation into the cells of the blood to carry oxygen, fight infections, clot the blood. Um, and rarely, or maybe not so rarely, one of these stem cells acquires a mutation at some point in life and grows at a faster rate than its normal neighbors. And at its basic level, this is clonal hematopoiesis. And in that first step, there's rarely any detectable impact on the function of the organ. The cells are apparently normal in number, um, and uh, and there's limited effect at the individual level. Clonal hematopoiesis in certain genes like DNMT3A have been linked, and TET2 have been linked with adverse cardiovascular outcomes, immune inflammatory signaling, um, and have a whole host of immune derangements that drive non-malignant pathology. And then on the other side, clones can acquire a sequential subclonal progression via additional gene mutations to develop a frank leukemia. And this leukemia um, um, uh, is associated with organ failure or cytopenias. Um, and so it's really this process that we're going to focus on today. There's been a lot of study over the past now 15 years or more about the spectrum of recurrently mutated genes in the development of myeloid malignancies. And these range from those that alter DNA methylation, chromatin organization, RNA splicing, DNA damage response, growth factor receptor signaling, myeloid transcription factors. They all share the common property of, in the right context, they drive clonal advantage or clonal dominance. Um, but it's important to recognize that they don't all do this in the same way, or in the same combination, or in random order. There's actually a highly stereotyped progression from initiation through intermediate stage of progression through to terminal transformation to a florid leukemia. And there are specific genes that are associated with each of these steps. And for orientation here, this is a very simple reductionist model of the kind of three large categories of myeloid leukemia. Those that have MDS-associated mutations, um, like those impacting RNA splicing and chromatin modification and one of the cohesins. P53 defines, in some cases, its own distinct ontogeny or type of disease. And then there's de novo AML, which is a little less degenerative in its origin and is more genetically simple. And the reason I present a reductionist model is because it's step one in trying to understand how these diseases progress, how they respond to treatment. Um, and I'll give you one example of that. At first blush, this model was simply classification, and some of its features have been incorporated into classification and prognostic models over the years, and uh, allow us to reframe or reclassify historical clinical trials, for example. And I'm just going to show you one example. And so in 2013 or 14, Celator and Jazz planned the phase 3 trial comparing CPX-351, a liposomal preparation of daunorubicin cytarabine, to standard infusional 7+3 for high-risk patients with secondary and therapy-related AML. This was based on clinical inclusion criteria defining high risk. And this was AML with myelodysplasia-related changes defined by recurrent cytogenetic abnormalities or a history of clinical MDS or CMML. And then The third group was those who had had a chemotherapy or cytotoxic therapy exposure. And this was according to the 2008 WHO criteria for this high-risk group of patients. And as you can see on the right, CPX-351, when analyzed in all these patients, was associated with an improved overall survival. And on this basis, the drug received FDA approval. In great shock and disappointment to the company, the approval indication evaporated shortly after they received it, meaning we started reclassifying disease using different terms. And so now there, the notion of biological disease or ontogeny is a greater explanatory model for disease behavior than the clinical observation. So these clinical groups can be reclassified based on genetics into some categories I'm showing you here, according to WHO and ICC classification, as well as ELN prognostic models. And so what does this trial look like when re-evaluated? Here are the three main inclusion groups. On the left, AML-MRC with prior MDS or CMML, so transforming after a chronic myeloid malignancy. In the middle, basically complex karyotype chromosomes 5, 7, 17 abnormalities, and on the right, post-cytotoxic therapy. And what we can see is that these are very heterogeneous groups when we look at genetics. Those with prior MDS or CMML were largely had MDS-associated mutations, with about 20% with P53 mutations. 15% or so looked like they had de novo AML. The AML-MRC with cytogenetics and therapy-related were each more than 50% P53, and then half something else. And so when we now reshuffle the deck to have more genetically concordant groups, this is how the outcomes look when we compare clinical classification and genetic classification from this phase 3 trial. On the left, these three clinical groups overlay, their KM curves overlap each other. When we use these four genetic groups, and I'm adding DDX41 as a recurrent germline contributor, we see a wide spread, even in this extremely high-risk group of patients. And so the genetic classification affords opportunity to better resolve outcomes in this group. So how does the drug look? Does the drug work differently in different types of disease? Here are the four categories now broken down by treatment arm. You can see in the bottom left, P53, no difference. And in fact, the bulk of the signal is within the group of patients who have AML-MR mutations. Those who have de novo disease, no difference. The numbers are really small here in the DDX group. There's a hint that maybe they respond better, but I'm going to leave that for future studies. But really quantitatively, this group here, AML-MR, drives and drove the clinical effect that was seen in the randomized trial. Even more so, what drives that benefit? It's really the ability to get to transplant. And so here I'm showing you on the left, no transplant, 7 and 3 versus CPX. The curves overlap and they're steeply down. And then here, 7+3 versus CPX among those who got to transplant. And so something about it, they didn't include the adequate correlatives to really explain why. But AML-MR who go to transplant actually have a shockingly favorable prognosis here for this group, who in the absence of transplant actually are largely dead by 1 year. And this holds true in a extensive multivariable model here. So treatment and transplant are both independently predictive of outcome among patients with AML-MR. So just P53, I have to speak to it very briefly. We looked at this group and said, is there any treatment effect? No, there wasn't. But we also we classified these based on empirical determination of allelic state by looking at NGS determination of copy neutral, loss of heterozygosity, or deletion of 17p in the presence of a mutation, or karyotypic chromosome 17 alteration, or the presence of two mutations 2-point mutations and categorize them as p53 single or p53 multi-hit. What you can see here is most were multi-hit, as is common in AML. Most of them were pure p53 disease, meaning they look like they started as p53, they progressed as p53, and that's true p53 ontogeny. There's this other small group, and most of the singles, which have subclonal progression of their MDS with a p53 mutation. And this in work I won't talk to you here about is these two types of p53 defined not only by allelic state, but by genetic context really drives clinical outcome. And here, it's really the allelic state and not the treatment. This is just looking at allelic state. So p53 single in this group of patients overlays with p53 absent, and multi is what drives the adverse effect. And there was no treatment effect among the multis. So allelic state in p53 induction outcomes. We also looked at here is because if you think about how do you want to apply a new therapy to the patient who's in front of you, you need to know its effect and you need to know its consequence to the patient outside of its on-target effect. And so we looked here on the left is among patients who got into CR, their time to count recovery to an ANC of 500. On the right, count recovery to a platelet count of 50 among those who had CR. And here's just days from induction with a dotted line as 28 days. And here I'm showing you each genetic group paired with their 7+3 on top and CPX next to it. And we can see for both ANC and platelet recovery, CPX, independent of genetic subgroup, was associated with longer time to count recovery. But that was particularly evident in the AML-MR patients who had poorly functioning marrow. And also, I think importantly, extended in these DDX41 patients, and then these de novo patients down here. And the last thing I'll show you about this trial is now if we use these genetic subgroups to now define early mortality, it's sobering to me. And so DDX41, they all survived the first 60 days. And then in the de novo AML and the AML-MR patients, they were about 15 to 18% 60-day mortality. And then the p53s, the multis had a greater than 25% 60-day mortality. And so if you, we can kind of cogitate on that for a moment. So high-risk group of patients and a lot of early mortality even among the different groups. And so conclusions from this is when we reclassify clinical history using biologically driven genetic subgroups, or the other way, genetically driven biologic subgroups, you tell me. AML-MR drives the benefit of CPX 351 over 7+3. And this effect was mediated by transplant consolidation. For p53, there's no difference in treatment, and allelic status is the main thing. But really, I'd ask you, should these patients get intensive induction? This is a side conversation we can talk about for a while. More than 25% early mortality, no real benefit. So I think important to think about. And then there's delayed count recovery that we should be aware of, particularly among the AML-MR patients who achieve CR. So this sort of approach now of reclassification is being applied to aza-ven and novel therapeutics for AML. And there are different response characteristics and toxicity characteristics on this very simple reductionist categorization of disease. And so I just wanted to highlight that as an example. It's kind of like an old story that is now kind of coming forward. And I want to move to into some different space, which is really trying to understand how the germline state modifies the risk of developing myeloid malignancy, but also modifies the path to malignancy. And so what I'll talk about um, is the initiation of clonal myeloid disease and the concept that somatic clones are selected um, by fitness constraints that are defined by the germline, that are defined by aging and exposures. And so here in the germline encoded fitness, here's a model, and I'll return to this a little bit. In this middle band is normal fitness, and the clonal dominance is really measured by the fitness of a clone relative to whatever the baseline is for that bone marrow. And so in marrow failure syndromes or predispositions, many times that baseline fitness is much lower. And so there are many ways where a somatic mutation can augment the fitness relative to the background, but not all of them drive leukemia. And what I'll describe is the difference between those paths. And then this is now looking at the uh, fitness over time. And this happens in everyone. There's a gradual loss of fitness that occurs with aging, and that creates a novel selection pressure with aging or a fitness constraint with aging that allows for the selection of clones. And so the hypothesis that we go into with this is that the age and gene distribution of clonal hematopoiesis in any given clinical scenario reflects the mechanism and the magnitude of fitness constraint. So that composite of how is the fitness constraint defined and how big is that constraint or how tight is that constraint really influences the initiation of clonal disease. And so for this inherited marrow failure state, baseline fitness is uniformly low in the ones I'll talk about. And we see a lot of clonal metapoiesis. And there are questions that arise. Is this neutral drift because you have a reduced pool of stem cells? So you just have a less complexity and then neutral drift. Are there disease-specific factors that drive selection? Does the path or the type of CH that or clonal metapoiesis that we see, does that determine or drive the leukemia predisposition? And can we use a rational understanding of mechanisms of leukemogenesis and mechanisms of fitness constraint to develop a rational surveillance strategy for patients with ultra high risk of progression? And can that rational surveillance strategy be the basis for preemptive clinical intervention to mitigate the risks that are associated with transformation? And so here's the paradigm that I'll circle back to. I just want to put it in your head to start with. And so again, just to beat the dead horse, baseline fitness is low and uniformly low in many of these marrow failure syndromes. Anything that can normalize that defect will cause a clone to grow better than its baseline impaired neighbor. And then there's a subset of mutations which may mediate transformation through loss of fitness sensing, through bypassing tumor suppression mechanisms. And unlike the normalization, where the tumor suppression mechanisms are still intact, on the right, tumor suppression mechanisms are lost and leukemia ensues. And so, how did we go about evaluating this paradigm? And so, this really rose out of I would say, serendipitous discovery, which is we looked at a whole bunch of MDS patients from 6 months old through 75 years old, 77, who had allo transplant. And then we looked at those characteristics that are genetic characteristics that are associated with age in MDS. And so over here, you see the classic splicing factor P53, DNMT3A, TET2, older age, younger age, acquired predisposition, PIGA, GATA2, and biallelic SBDS mutations. When we look at outcome among these three groups, those with PIGA or GATA2, they do really well. Those with SBDS mutations, they do really poorly. And this has been now replicated in other studies. So SPDS, for some reason, is associated with very poor outcomes. And I'll cut to the chase. It's because they all have p53 mutations. So that is what drives leukemiogenesis, and that's what drives poor outcomes in leukemia and SDS patients. Most of you may not be familiar with Shwachman-Diamond syndrome. It's a disease of impaired translation that's mediated by a ribosome maturation defect. And so normally the um, the nascent ribosomal components of 60S and 40S are in the nucleolus. They're exported to the cytoplasm. 60S is maintained in an unjoined state by binding of EIF6, which is an anti-association factor. Um, SPDS, the gene that's mutated in Shwachman-Diamond syndrome, uh, works with EFL1, a GTPase, to kick EIF6 off the nascent 60S, allowing it to join the 40S to create the mature, translationally active ADS ribosome. That's how it's normally regulated. Uh, uh, Stepwise ribosome joining and translation. When you lose SPDS, you lose the ability to kick EIF6 off the nascent 60S, and you get a accumulation of these free 60S EIF6 um, um, molecules in the cytoplasm, and you have a reduction in the overall abundance of ADS translationally active ribosomes, and a downstream reduction in protein translation, which activates p53, which drives bone marrow failure. You can get a sense here, this is going to be a tight fitness constraint on cellular growth if you can't translate and you're activating p53 at baseline. And so that's what happens. These patients have short stature, exocrine pancreatic insufficiency, and a remarkably high risk of developing leukemia, oftentimes in the teens and 20s, but now as we're learning, even later. And so to define um, the somatic pathways of progression, we did some discovery sequencing in collaboration with the Shwachman-Diamond syndrome registry in patients with and without leukemia. We identified somatic recurrent mutations and then used duplex ultrasensitive sequencing in a validation cohort of 327 samples from 110 patients. Um, don't ever tell a pediatric marrow failure patient that's doctor that's not a lot of patients. This is a huge effort by Akiko Shimomura and the SDS registry to accumulate these serial samples from these patients over a long period of time. And so this is a large group of SDS patients. All the leukemias had p53 mutations. I already said that. Those without leukemia, 70% of them had clones. Remember, these are young patients. So how does this compare to regular age-associated sporadic CH? Um, Age-associated CH had largely DNMT3 and TET2, single mutation, much older age, 60s, 70s, 80s. SDS clone is something totally different. It's ubiquitous by adulthood. So by 21 here, mind you, there's not that many patients, but they all had CH. But even in the teenagers, we're talking 90% had CH. And even in the first decade of life, more than half had detectable CH. What was that CH? It was EIF6, never been seen to be mutated in humans before, p53, PRPF8, casein kinase. Um, None of, or barely any of these DNMT3 had TET2. So it's not the same CH. And it's not one mutation. Some of these patients had 5, 10, 15 different mutations that are detected. And these are young patients. So it's different. Something's different here. And so this high frequency of EIF6 mutations raised the possibility that maybe disrupting this EIF6 RPL23 or nascent 60S interaction in some ways promoted ribosome joining and growth of the cell. So here's what it looks like. EIF6 is this little tiny cap right here that binds exactly to the site that the 40S binds. And so it's just inhibits 40S binding through steric hindrance. And when you rotate it out, it's got this beautiful little like kind of claw. It's got bifold symmetry with a little binding area right in the middle here. And so there are potential mechanisms that we imagined. So we could have mutations that disrupt that binding interaction, that protein-protein interaction, or it could just be stoichiometry. You knock out the EF6, you cause haploinsufficiency, and you favor ribosome joining just by the relative concentration of EF6. So there was something like 250 different EF6 mutations, or different, um, not different mutations themselves, but mutations combined with patients in this cohort. And many of them were missense substitutions. Those are on top, with two very recurrent alterations here, R96W and N106S. And then also scattered truncating mutations. These were splice site mutations, frame shifts, nonsense mutations. These missense substitutions were almost uniformly in key structural, secondary structure regions as well. And so to kind of imagine or predict what these might do, we developed a homology model of EI6 based on a bunch of the published structures in yeast and archaea, and then mapped each mutation onto that homology model and predicted the impact on a bunch of characteristics. And so here, that recurrent R96W, here's what it looks like. Normally, the arginine 96 and asparagine 78 are really in close proximity here in black. These are two hydrogen bonds that kind of keep that together. When you mutate the arginine to a tryptophan, those hydrogen bonds break, that connection falls apart. And as a result, there's a large change in the free energy, resulting in protein destabilization. And so when we express this mutant at high levels, there's no protein. So it's a protein destabilizing mutation by disrupting these hydrogen bonds. And so is that how all these work? So we mapped these all, calculated their delta delta G's, the free energy changes, and it's hard to see, but over here on the right is kind of like the take-home message. Over here is a message showing you that we express them, and here is the Western blot showing that most of these mutations with high free energy changes are destabilized. And so this is just a missense mechanism for resulting in a knockout of that allele. And we included this one as a teaser. So N106S, if you remember, was this highly recurrent mutation. So when we look at that, it's situated right at that interface between EF6 and RPL23. And it's these interface mutations here, when we look at the free energy change, they have minimal impact on protein stability or predicted impact. And all the other missense substitutions have high impact. So here are your destabilizing mutations. And then there's this site, which we hypothesize would not destabilize the protein, but would destabilize the protein-protein interaction, destabilize its anti-association function, kind of make it fall off. And so these interface mutations here have actual increased energy of binding as predicted when we model that RPL23 AF6 interface. And so here is just the same thing where we can show that this N106S mutation is predicted to make that interaction fall apart. And so to really prove it, though, we use sucrose gradient centrifugation and Western blot to really see what the impact of this mutation was on ribosome joining. And so the take home here is usually there's some of this free EF6, but that EF6 is usually bound to the 60S and then gone from the 80S. When we express the mutant, all of that mutant is in that free fraction. None of it is bound to the 60S. Only the endogenous here is bound to the 60S. And so this kind of nails the mechanism there that this mutation simply breaks apart that interaction. And this is supposed to say M106. And what you see is you see increased ADS formation. And so here's the summary from this stuff, which is um, the two most frequently mutated genes. EF6 mutations repair the ribosome defect. They improve protein translation, and in so doing, decrease p53 pathway activation, here reflected by CDKN1A expression. P53 mutations fail to repair the ribosome defect, fail to improve translation, but obviously still knock out p53 function. And so together, they both result in the same end, but one fixes and one doesn't fix the underlying issue. So these are frequently found in the same patient. Um, so you could say maybe they have a cooperative function. Maybe they are in classic cancer genetics model where you have one that enables the other one to stick. Or maybe this is just parallel selection in a field of dysfunction. There's different shots on goal of clonal outgrowth. And so we did some single cell sequencing in patients with many mutations, and we're able to prove that these are all parallel clones. So independent genetic events developing within this dysfunctional marrow. Here, these are individual mutations in columns, individual clones or cells in rows. And so this is a patient, for example, with 9 different clones, all separate genetically. When we look over time, most of these mutations are stable. They don't do anything, even the p53 mutations. And so having a p53 mutation doesn't tell you you're imminently transforming. And this will resonate if you've ever seen p53 clonal hematopoiesis. What does matter is all the leukemias had biallelic inactivation through LOH of various sorts or biallelic point mutations. And we can even take a patient who developed one of those leukemias and track back 6 years of their serial samples and identify among these 13 clones that they had, identify that moment 5 years before their transformation when they got their 0.1% biallelic. They lost their heterozygosity, and that then started to grow. This is on log scale, started to just grow, while all the other ones were stable. And at this last interval, just blasted off. And so maybe that's a mechanism or pathway for rational surveillance. We can identify incipient transformation by identifying at-risk clones within this C. We try to balance when to deploy transplant. Because if any of you have ever transplanted someone, there are risks of commission there when you think about toxicities. And so this is just reiterating the model. EF6 mutations repair the defect, but don't allow for transformation. P53 mutation, monoallelic, allow for increased growth, but not transformation. And then the biallelic hits drive leukemia. So is this a generalizable model? Here's another patient. This was a kind of a weird one where we were doing some sequencing. And we found a patient with MDS, 70 years old, no family history, had a P53 mutation and a variant of uncertain significance in TERT. Patient also had short-ish telomeres. Couldn't be 70 years old with no family history, but a germline telomere disease. And so we looked into this a little bit more. TERT it has extremely high degree of constraint for missense substitutions or predicted loss of function mutations in the genome. So it's a very tightly constrained gene. Here is among all genes, and here is among even telomere-associated genes. When it's mutated, the textbooks tell us that there's a mucocutaneous triad of oral leukoplakia, nail dystrophy. There's also bone marrow failure and exceedingly short telomeres. And this is not this patient. So we looked in these in MDS patients, sequenced all of the telomere-related genes, identified all the variants, split them into common and rare with the hypothesis that rare variants might have snuck through evolution and be driving some telomere maintenance defect. So in MDS patients, the TERT rare variants, TERK and DKC1, were associated with short telomeres relative to others. And when we looked at these candidate mutations, functionally, we cloned them all and tested them. 90% of them or so were actually impaired, many of them severely impaired, um, um, suggesting that there's a germline telomere dysfunction that's driving MDS biology. Did this matter for patients? Yes. Patients with TERT rare variants had reduced overall survival. This wasn't due to relapse. It was all due to toxicity. And it was exactly what you would expect. Non-infectious pulmonary disease, things like that, that are associated with telomere disease. And when we look at even more resolution, it's those who get intensive conditioning, myeloablative conditioning, who really have that jump in NRM, and this is including the TERT and TERCs. But here we're looking at more than 50% non-relapse mortality with ablative conditioning with a TERT or TERC rare variant in MDS adults, not with TBDs. These are not known to have telomere disease. So we incorporated this into our standard panel sequencing back in 2019. So all patients with hematologic abnormalities at Dana-Farber will get this test. And we've, I think, identified since then about 109 patients with TERT rare variants among patients with largely myeloid malignancies. Few of them are pathogenic or likely pathogenic by ACGME criteria, and most of them are VUSs. And so most of them go in, and the clinician will say, okay, I don't know what this means, and just watch. Um, so we measured the telomere length in all of these patients by flow fish, and sure enough, they have short telomeres. We measured the functional impact of all of these variants. Many of them have severely impaired telomere extension here in this kind of red, pink area, intermediate here in white, or some of them were wild type, as expected in blue. It's really raised the question of just how broad is this observation? So how common are TERT rare variants, and how much explanatory power might they have for MDS and AML, or even cancer more broadly? Um, and so we here, this is just looking at, UK Biobank, all of us, Nomad, showing the distribution of these rare variants or all variants. Most of them are only detected in one or two individuals. They're across all domains, but uh, and 96% of them are VUSs. And so that's why we never hear about them, talk about them, think about them, is because they're VUSs. And that's why no one associates TERT with, until recently, with some of these things. And so we decided to, I think we identified something with, lately, I think we're up to 1,700 TERT rare variants across all human population data sets. So we've cloned them all and developed a arrayed strategy for functional testing, where we do 5 biological replicates for all of them, express them inducibly in cells, and then measure telomere length. This is just validation using standard kind of Southern blot. Uh, um, just in some. I'm not evil. um, um, qPCR is really the scalable method by which we do that. And so here's validation. These are bona fide telomere disease in the telomerase database. Here's how they perform. And so here are the controls, and here are the database samples. We actually do better than the standard TRAP assay, in part because we can identify enzymatically capable TERT that has defects in localization or other mechanisms of function. And so many variants in TERT are shown to be wild type in the standard assay, even when they don't work in cells because they can't get to the right place. And so um, here is just, this was a data grab from a while back, looking at domain specificity of these variants when we look by, oops, when we look across the VUSs. Linker, this is an unstructured linker, doesn't do anything, and consistent with that, they're all wild type. And then there's tons of dysfunctional TERT in these rare variants or ultra rare variants. And here I'm just showing you the tools that we use clinically to deconvolute VUSs are really poor. So these are our best current, like the current best deep learning models for variant effect prediction. Eve uses evolutionary conservation to predict benign, uncertain, or pathogenic based on scores. Alpha missense uses AlphaFold predicted structures to identify or to categorize. What you can see here is these are the benign, intermediate, or pathogenic based on each of these prediction models. And then on the y-axis is their functional score. And you can see that the pathogenic is pretty specific. Um, Intermediate is across the board. And then the benign predictions, this is the real fault of these prediction models. Many of them are actually functionally impaired, including severely impaired or dead in the assay. And so um, we kind of dig into this and say, why might this be? So I'm focusing on this 10 domain, which is at the N-terminal domain of TERT. It's in the first 180 amino acids. It's important for binding to TPP1, which is how TERT finds the right spot on the end of DNA to act, to extend telomeres. And I'm just showing you here, this is missense tolerance ratio. So this is kind of areas of the that are the least tolerant to missense substitutions in evolution. And you can see that these severely impaired variants are really centered on that intolerant area, um, and that more than half of the variants are dead. These are just regular people walking around. Half of them have dead telomerase because of this defect. And why don't these things work in the models? Is because I think because the 10 domain mediates intermolecular interactions and intramolecular interactions that are poorly predicted by structure. And so those are um, um, 10 domain TPP1 interactions and interactions with the template, the Turk, the RNA template, among others. And so um, digging into it a little bit more, we've taken a look now at multi-allelic residues that have divergent um, uh, functional effects. And so here are just three examples um, at C76, G135, and R1086, where one variant is dead and the other is preserved or wild type, saying maybe this will afford us some insight into where the models fail and why these variants have um, these effects. And so here are these variants according to their alpha missense in Eve, most of them benign. And so this is a point of failure for the models. So that's why we're digging into these. We use molecular dynamics simulation which is essentially like a computational approach that uses basic rules of physics and and molecular function to not take static pictures like you would see in a crystal structure but predict how molecules interact over time iteratively. And so what we can see here is that if we look at this, this is the G135 result. The G135R here, it actually forms a salt bridge without altering the conformation. Um, and as a result, it actually has slightly better contacts with TPP1. So it actually performs well, if not better, than wild type. Whereas this G135E um, incurs electrostatic repulsion because it's rated in like a acidic patch of TPP1. And so it drives decreased contact and breaks that recruitment interaction. And so we're now using this approach to build a new deep learning model for variant effect prediction that incorporates these structure function findings that we have. And I'll skip that for now. And so moving to the last little bit, let's connect it back to disease initiation. So that's setting the stage of that was a rabbit hole. Let's just call it that, call it what it is. It was a rabbit hole for us because we were confronted with VUSs. And if any of you ever have to deal with VUSs, you know that they should inspire loathing and frustration because we know that they either are one thing or another. And when they're pathogenic and you don't know that, or you only know it retrospectively, the implications for patients can be severe. So these are patients, if you take them to a myeloablative allo, you're incurring, as I showed you before, a 60% chance of non-relapse mortality. And so this is meaningful. And so that was the rabbit hole. Um, um, Hopefully the postdocs like the rabbit hole. But here's getting back to the disease initiation. If we say aging is associated with decreased fitness, there are multiple ways that aging could do that, one of which could be the cumulative effects of replication. And so stem cells undergo telomere attrition as they replenish the blood. And so maybe this is a fitness constraint that evolves over age and could explain the age-associated risk of leukemia in a subset of the population. And so here's what the fitness would look like. And here's, if you have that fitness defect, you could select out clones there. And so sure enough, um, clonal hematopoiesis in patients in our telomere disease program, um, they have a very different spectrum of CH than healthy donors who are DNMT3 and TET2. We see lots of PPM1D, P53, U2F1, S34. When we do clonal decomposition with single cell, these are all, again, independent clones growing out. And then those that transform, the genetics are different than the baseline CH. It's P53 and U2F1 S34F, which drive transformation in patients with telomere disease. And there's no PPM1D. And so maybe this is an EIF6 P53 analogy like with Schwachman. So what is PPM1D? It's basically the negative regulator of the entire DNA damage response. It's a phosphatase that that regulates ATM, check one, ATR, P53, check two, MDM2, gamma H2AX, everything. It's the return to normal that's required for regulation of the DDR. The mutations lop off a degron, resulting in a highly stabilized phosphatase that's hyperactive. And so this quiets down the DDR. And so what happens with telomere attrition, so with every replication, um, before S phase, the telomere is capped. It's kind of looped over itself. It has to uncap. In the process, it becomes deprotected um, and is at risk for being recognized as a double-stranded break or DNA damage. Telomerase sits on it, extends a few times, then it recaps, and then we're good. If in the setting of attrition, eventually it has a hard time actually recapping, and that state of vulnerability is extended, and DDR is activated above threshold. And so you get a ATM-dependent check2 P53 senescence program that activates G2 and G1S checkpoints and drives senescence. So you could imagine that mutations in ATM check2 PPM1D or P53 could in different ways attenuate these steps. And so we have developed models, and I'm not going to go into this, but where we engineer natural attrition that results in a stereotyped deprotection of telomeres. They activate that entire program, and this is CDKN1A. And so they go through this process. When they reach that point, they do the right things when it comes to cell cycle arrest or checkpoint activation. And we use that as a way to then model the effects of all these different mutations on the naturally occurring telomere deprotection. And I didn't correct this slide, but essentially when we compare PPM1D and P53, P53 mutations allow these cells to escape senescence, and so they just keep growing when they should stop. And PPM1D just extends it by a passage and a half. And so it delays the activation of P21, but is overwhelmable by continued DDR signaling. P53 actually activates a whole second kind of contingency pathway that I won't get into now. But we're digging into now, this connection between deprotection and clonal selection. And so how broad is this issue? This is when we look at the UK Biobank, we see many genes where their rare variants impact telomere length, either to shorten it or to lengthen it. And so this, as a paradigm of aging and cancer risk, is something that we're broadly engaged in now. And this cuts along whether these are affect the C strand of the telomere or the G strand. And this is essentially over here. There's a G-rich strand, which is where the telomerase stuff happens, and then this C-rich strand, when there are mutations in genes that regulate that, they actually cause aberrant lengthening. And so this is an area of future ongoing exploration. And so we're asking questions like, is the age-associated telomere attrition really a generalizable mechanism by which individuals have MDS risk? Do patients with telomere disease kind of accelerate that process? Is it different? And then as a teaser, I'll show you we've now developed and deployed ultra-sensitive duplex sequencing panel that's scalable now. So we can do studies in, I think we've done probably 12 to 13,000 samples now. But this is ultra sensitive. This is a level of detection down below 1.1% parent-to-allele fraction. You can see that mutations accumulate with age. We've done the negative controls that the infants are dead negative. But what you can see here is that here's the CH you know about, where we focus on DNMT3A, TET2, ASXL1. This is relatively young. And then there's this whole wave of DDR-CH that happens later, really raising the possibility that that's what happens. And so this is, I think, how CH works over time with evolving fitness. And so when we sequence, we take a slice at any one of these time points, and the CH that we see is the CH that fitness got us. And so that's what I have for you today. Chris Riley is really the partner who drove this when he was a postdoc in my lab and is now running a large telomere disease multidisciplinary program. So if you ever have variants, call him or email him. He will give you a comprehensive evaluation and refer patients to him. That's all he does. And so lots of collaborators, and thanks for your time. And I recognize I went to my time, so I'm happy to talk with anyone at any point. Yeah, thank you.
Found it interesting that you talked about chocolate diamond first and its canonical role for liposome, and then you went into telomeres. But there was a report a few years back showing that chocolate diamond protein might play a role in telomeres too. I wonder if you find that that's a possibility compatible with what you've shown, or
Everything that we see from so I view this sort of sequencing approach as a CRISPR screen essentially, and so the clonal hematopoiesis that we see growing out in Shwachman-Diamond syndrome is all related to things. So P53, and then everything else is about fixing ribosomal P53 activation. So translational stress. So we see mutations in dead box helicase proteins in RPL5, RPL22, all ribosome based. And I don't see ATM, CHEK2, PPM1D, which are now the signature of telomere stress. So I would say the humans tell us that that's not true. Like that's not the driving selection pressure. I should be more accurate. Does that make sense? Like, yeah, I like following the humans really just tell you what does happen through selection. I think that's the.
Yeah, great talk, Coleman, as usual. So for the TP53 in the Vixios study, like you, I was surprised with the 25% induction or 60-day mortality. So I understand this is with both drugs, the same 25%, or?
Yeah, it was, I didn't say that, but the early mortality was the same across molecular subtypes, irrespective of drug.
So is your sense that those patients are dying sooner? Because, I guess the other part of the question is that this study was 60 years and older. So the first question is, do you think this applies even for younger patients with TP53? And the main question is, do you think that this is
Like a very
driven?
optimistic clinician. I love you, Amr.
Yeah. And do you think this is uh, a reflection of primary induction failure and fungal infection and
Yeah, it's going to
all
be
that,
a.
or is it like some excessive mortality, chemo-related toxicity that we just don't
I think it's a composite.
recognize?
I think it's a composite. I will say that there's a strong association, and I'm not going to get into this, but between albumin less than 3 and early mortality generally, but then in this trial for sure. And so I think, what does that say about a patient? You tell me, but that tells us maybe it's not just about induction failure, but there's something about the host there that might influence, but we don't have the data to deconvolute that.
Thanks.
Thank you for that great talk. You did show the TERT mutations, the non-relapse mortality is 50%. I would lose my job. What's driving it?
Um, so cause of death in transplant is tough to ascertain oftentimes, but there is a markedly increased risk of non-infectious pulmonary disease. There's also a more of a consequence of acute gut GVHD. And so there's no difference in GVHD, the cumulative incidence of GVHD, but the consequences of severe gut GVHD are more in patients with short telomeres. You can imagine because they have less regenerative potential after injury of the gut mucosa. We have mouse models actually to show that as well. If you induce GVHD in a mouse, they with short telomeres they die of gut failure. So that I think those types of things are what they die of.
Transplant them early, but then you show the other side of the story.
Well, I guess it's mode of transplant. So I would would not do an ablative transplant in these patients and maybe reduce intensity or other like a toxicity sparing strategy would be better. Yeah.
I have a practical question.
Sure.
To detect short telomeres, how confident can we be from our flow fish result? And is there a commercial PCR assay that would be appropriate for use?
So the best assay that I know of that we use is the repeat diagnostics flow fish telomere length testing. What I will say is that as you age, the normal physiologic telomere length approaches pathologic telomere length. And so the test in terms of age adjusted percentile becomes meaningless when you're non-pediatric because all the kids have very long telomeres. And so they separate out normal and pathologic. But as you go across time, pathologic remains the same because the cell doesn't care whether you're 5 years old or 80 years old.
Right.
Short telomere. But then aging brings it down ubiquitously.
So it's not a very informative test in
So it's not
adults.
sensitive. So you can definitely have, if you take a genotype first approach, you can definitely have people with dysfunctional telomere maintenance who have normal appearing telomeres, although they tend to be less than 50th percentile by age or less than like 4.5 kb or something like that. But remember, flowfish is also population level, like cell population or telomere population. It's single cell flow. But what matters is how many short telomeres you have within a cell, and there's a lot of heterogeneity. We can get into that later, but so
Yeah.
I
It
think
sounds like a genetic
right now
approach
we're
for
taking a genotype first approach as the initial. Okay, thanks everyone.